Hello, my dear students. Welcome to my today's class, that is one chapter pertaining to historical embryology. I am Dr. MEERA B.K, Associate Professor and Head, Department of Zoology, Maharani Science College for Women, Palace Road, Bangalore 1. Here you can see my mobile number on this slide, so also my email ID. After the talk, any queries to be sought or any questions, any discussions, don't hesitate to contact me over either my cell phone or through my email ID. Let us start the lecture now. Developmental biology, earlier it was called embryology, is the study of development of embryo. It is a specialized branch of biological sciences dealing with the development of an individual organism. In earlier days, this branch was called embryology. Embryos means embryo, logos means to science, to discourse, that is the study of embryos. But with the wider perspective, in the later years, embryology came to be known as developmental biology. This branch has several sub-branches like historical embryology, descriptive embryology, comparative embryology, experimental embryology, chemical embryology, and analytical embryology, that is today's modern developmental biology. Today what I am going to discuss is one chapter under historical embryology. Before going into the historical aspects as such, we shall understand the scope of developmental biology after knowing its definition. Developmental biology serves as an important tool for analyzing problems pertaining to cell physiology, cytogenetics, and evolution. Then, the phylogenetic relationships among different animal groups can be better understood by seeking embryological evidences. Then problems pertaining to cell growth and differentiation, cell repair and regeneration can be analyzed meticulously with the background of developmental biology. Then the applied aspects of developmental biology find an important place in medical sciences, especially in obstetrics and pediatrics. Further, development of modern birth control techniques is possible today because of basic understanding of development biology. And recently, we study what is known as MEERA, that is medically assisted reproductive techniques, both in humans and animals. For this, classical embryology forms the basis. So, I have briefed the scope of developmental biology. Now, let us understand the theories of development, a historical review. As I told you, this is a part of historical embryology. A brief survey of history of development reveals the two possibilities of development of an ovum or egg. One view believes that embryo is already present in one of the gametes. This forms the basis of the theory of preformation. And the other one believes that preformed embryo is absent in the ovum but somehow appears during development. And this forms the basis for the theory of epigenesis. Now let us study the preformation theory. In 17th century, a Dutch scientist by name SWAMMERDAM advocated that gametes contained preformed embryos or miniatures of the adult. He and many other biologists of that age like Haller, BONNET, SPALLANZANI and MALPIGHI thought that the ovum contained transparent, highly folded, immature and unobservable miniature of the adult which in some way was stimulated to growth by the seminal fluid. And those scientists which believed that the miniature of an individual is present in the female gamete, that is ovum, were called ovists. Then Leeuwenhoek, Hartzacker, etc., another group of scientists, believed that preformed miniature embryo is present in the head of the sperm and not in the egg, and that miniature was called homunculus or animalcule, meaning spermatozoon. These scientists who believed that preformed miniature embryo is present in the male gamete, that is sperm, were called animalculists or spermists, in contrast to ovists or homunculists. These spermists considered the sperm as the seed and the egg as the soil in which the seed is planted. Further, few ovists like SWAMMERDAM and BONNET went to an extreme to advocate what is known as encasement theory or embodiment theory, which hosts that successive generations of individual organisms pre-existed one inside the other in the germ cells of the mother. They also estimated that as many as 200 million years of human beings were present, already delineated in the ovaries of Eve. All these cock and bull stories persisted very well till in 1759 an embryologist called CASPAR FRIEDRICH WOLFF offered experimental evidence that no preformed embryos existed in the egg of chicken. He suggested epigenetic manner of development, which came to be known as epigenetic theory. And today it is a well-known fact that epigenesis is more nearer to the truth compared to the theory of preformation. Now let us see what the epigenetic theory says. Wolff suggested that future embryonic regions of an egg are earmarked, and these regions he called granules or globules. And these granules lack in any arrangement that can be related directly to the structure of the future embryo. And gradually the globules or granules organize into rudiments, which in turn took the characters of the embryo. This method of progressive development from the simpler to the complex through the utilization of building units is called epigenesis. Another developmental biologist or embryologist, as we call, by name Carl Ernst von Baer, he proposed that he confirmed, in fact, the epigenetic theory in 19th century. Carl Ernst von Baer, even today, is treated as the father of modern embryology. And Hans Driesch, in the year 1900, through his experiments on sea urchin, proved that epigenesis is a fact compared to preformation. What he did was, the blastomeres of sea urchin were separated at a two-celled stage, and to his surprise, it was found that a complete embryo was obtained out of each of the separated cells. That means epigenesis is more nearer to the truth, as I have already told you, than preformation. If preformation theory were to be true, instead of getting an entire embryo, a half egg could produce only half an embryo and never the entire. Then, Baer proposed his law pertaining to the theory of development. He was, in fact, the first embryologist to document all the available embryological data in a coherent form. Then he made landmark investigations and made certain important generalizations which are considered true even today. His most important contribution is what is known as Baer's law. The Baer's law states that more general features that are common to all the members of a group of animals are, in the embryo, developed earlier than the more special features which distinguish the various members of the group. You will understand the statement better if you listen to the explanation. What exactly the law means is that features that characterize all vertebrates, such as brain, spinal cord, axial skeleton in the form of notochord, segmental muscle, aortic arches, are developed earlier than the features distinguishing various classes of vertebrates, such as limbs in quadrupods, like amphibians, reptiles, hair in mammals, feathers in birds, etc. Sorry for the spelling mistake. It is not features, it is feathers. Then, the characters distinguishing the families, genera, and species come last in the development of the individual. Thus, the early embryo has a structure common to all members of a large group of animal kingdom and may said to represent the basic plan of organization of that particular group. This law was formulated before the advocation of the evolutionary theory. Later, therefore, in the light of evolutionary theory, the law was reinterpreted by two more developmental biologists by name Muller and Haeckel in the year 1864, and they named it as biogenetic law. This biogenetic law is also called recapitulation theory. The theory states that ontogeny is a recapitulation of phylogeny. And just like the earlier case, you will understand the statement better once if you listen to the explanation. What exactly the statement means is, Features of ancient origin appear early in development, while those of newer origin develop late. Hence, ontogenetic development, which exactly means stages in the life history of an organism, that is developmental history, presents the various features of the animal's organization in the same sequence as they evolved during the phylogenetic development. So, ontogeny means developmental history, phylogeny means evolutionary history. Then, gradual transformation that have taken place during the long period of evolution are reproduced, although very briefly, in the development of an individual animal. According to this, chick embryo should look like a fish, then it should look like an amphibian, then it should look like a avian embryo, and so on, which normally does not happen in the actual development. The chick embryo resembles that of a fish in the earlier stages, but then looks like a reptilian embryo. That means the early stages of embryos of fishes, amphibians, reptiles, birds, and mammals look somewhat similar. When we look at this explanation and what exactly happens during the development, I feel the law can be restated and modified this way. Instead of putting it as ontogeny repeats phylogeny, we can just be more clear like this. Ontogeny of an animal repeats the fundamental steps of the ontogeny of the ancestral forms. My dear students, thank you for your attention. Stay blessed. Any queries, do contact me over my cell phone or through my email ID. Thank you.